this is a really unique and exciting opportunity for me to move away from a scientific talk to a more personal experience talk, where literally I had an experience of moving from the laboratory bench to my mother's bedside, and a story about how her genomic data changed her cancer story. Beginning of the precision medicine era began with Brian Drucker and others bringing Gleevec to the clinic, where it was the first example of how targeted therapy could actually have effectiveness in cancer. And specifically, using a targeted therapy, Gleevec, against a gene fusion, BCR-ABL, in CML. Since then, many laboratories are using this genomic information and functional data, combining with drug screens and proteomics to rapidly advance the field and bring precision medicine to even more genomically complex cancers. This is when I decided to join Brian's lab in Inspiration 3.5 years ago, working on AML, the Beat AML project, where we're trying to characterize novel mutations in AML. However, this became all too personal when in 2014 my own mother was diagnosed with intrahepatic cholangiocarcinoma, stage 4 in fact, just shortly after this photo was taken. It was a very hard diagnosis. This is a rare cancer, it's an aggressive cancer, and many of these patients present late in the clinic because of their no symptoms, and so they present with late stage and poor prognosis, about 3 to 6 months to live. I finally understood what all these patients were feeling, a sense of anger and frustration and uncertainty of how even to proceed. But with my knowledge of cancer and treatment, I knew I had to get her into one of the best cancer institutes, and she started her standard of care, chemotherapy. Now, this is the only time I ever saw my mom really sick. She was fine before, and it was difficult to watch her experience these side effects. Most of all because it led to no measurable effect, as many of these cancer cholangio patients do. So I didn't give up hope, because I knew in my field that there is other options, luckily. And so I commenced on trying to figure out what the next steps were. I said, okay, we need to get sequencing of her tumor. And I was really surprised to find out that this was not standard of care at this institution, that in fact I had to ask for it. And so I did, and then luckily for her, this should have been a black background. Luckily for her, we did find a mutation shown there, and it's in the IDH1 gene, and it's a common mutation, and it's actually found in AML patients. And so I knew about this mutation, and I knew there were clinical trials. So this gave me hope. But I was so surprised when trying to navigate this system personally with my mother that we ran into so many hurdles, so many no's along the way. The first no. There is no clinical trial for your mom's type of cancer, only leukemias. And he said, you know, we should go ahead and go on with chemotherapy, because that's your best chance. I wasn't going to see that again, because she was so sick and we didn't have an outcome. And I said, no, I want to try this targeted therapy first. And I went to research where I could find a clinical trial. And in fact, there were these early stage clinical trials in IDH1 inhibitors. So I went to proceed to try to enroll her in these clinical trials. I contacted the pharmaceutical company, I contacted the principal investigator running some of these trials, and even though they were enthusiastic about my mother's story, they said, I'm sorry, there's no slots right now. These things, the phase 1 clinical trials obviously only open up so many slots at a time, and you know, it's gonna take weeks, months, which my mother obviously did not have. So I was desperate, and so we continued on persisting on, and miraculously a slot opened. And so we had to travel to the clinical trial. We went up there to enroll her. She failed the physical requirements. And we thought, oh no, her liver function is poor. You can't get on this clinical trial. Well, her primary tumor was in her liver, so I wasn't surprised about that. But she had overall amazing health, and I knew this was the molecular driver that was responsible for her cancer, so I persisted on. We actually petitioned the clinical trial, and they did eventually let her on. So here we are in Mexico, my mother climbing Mayan Temple, and that's how it felt, receiving finally this one yes after so many no's and changing her treatment forever. It felt like we had to climb Mayan temples to get in this trial. But you know what? It was worth it because she is now in this trial still, and she has a great relationship with her clinician there, and this phase 1 clinical trial has been amazing for her. In fact, she's one of the best responders in this clinical trial of her type of cancer. She's exceeded expectations. She's on this trial for 1.5 years now, and with completely stable disease. And the best part is without any kind of huge side effects. She's been able to live life normally, and she actually moved up with me now and is living with me, so this is great. Now, this experience has changed my life forever, and now I really see that the barriers aren't in the laboratory. I mean, obviously we need to gather more information about driver mutations, but I thought this experience is showing me that the data is not getting to the patient. The real hurdle is getting the data to the patient and being able to use it. And so I sought out a new career path, and now I'm working with Chris Corliss in the Night Diagnostics Lab, the director of the Night Diagnostics Lab, running a trial on whole exome sequencing of advanced cancer patients. Here we enroll patients, and we sequence their tumor, and we provide clinically significant information about the mutations that we find both in the tumor and germline, and then I handpick clinical trials here based on their tumor status, their mutational status, and combination of those mutations, and also immunotherapies if that's appropriate. So I'm finding here, working with these patients, that they face many tribulations using this data in addition to what my mom faced. For example, if the clinician isn't really able to discuss these complicated results in the clinic. If they're not able to move past standard of care, maybe they have to go through several lines of standard care before they can ever even address these results. And finally, if they can actually use the results to get on a clinical trial, having to travel, or getting an FDA-approved drug, or an off-label requires insurance to pay, requires time for the clinician to do that. So there's so many hurdles to come, but I found being able to contribute to patients in this way has changed my life. And this is the first step, and I hope that we can have many more patients have this type of story, especially with having panels like this where we can find new ways for patients to use and access their data. Thank you.